So now I will be dealing with plasmid. Term plasmid was coined by Lederberg, and his discovery of plasmid is credited to the Tatum. Now what exactly this plasmid is? Now let's say this is a bacterial cell. In the bacterial cell, this is main DNA. Main DNA, that is chromosomal DNA, that is nucleoid. Right? This is main DNA. Main DNA is double-stranded, but here I am showing it as single-stranded for the purpose of simplicity. Secondly, main DNA is highly coiled. But again, for the simplicity, I am showing it in a simple, uncoiled circular manner. Main DNA के अलावा बैक्टीरिया के अंदर there is also presence of extra genetic elements. Main chromosomal DNA के अलावा बैक्टीरिया के अंदर there is presence of extra genetic elements. This extra DNA which is present inside the bacteria along with the main DNA, it is known as plasmid. So what is plasmid? Plasmid is extra chromosomal genetic element. Extra chromosomal genetic element or extra DNA, this is going to represent plasmid. Again, plasmid is dsDNA. What do you mean by ds? Double-stranded. And it is circular as well as coiled. Double-stranded है, circular है और coiled. Same condition, just like the main DNA. Now this plasmid, the size of the plasmid is smaller than the main DNA. Means the number of genes which are associated with the plasmid, they are less. Size of plasmid is smaller than the main DNA. That's why plasmid is also known as mini chromosome. Size is small. Number of genes, they are less. That's why they can also be known as mini chromosomes. Now this plasmid, it has the ability to show replication, and after the replication it forms several copies. प्लाज्मिड रेप्लिकेशन शो करेगा और अपनी बहुत सारी कॉपी फॉर्म कर सकता है. बट फॉर द पर्पस ऑफ रेप्लिकेशन इट इज नॉट डिपेंडेंट अपॉन मेन डीएनए. रेप्लिकेशन पर्पस के लिए मेन डीएनए पर डिपेंडेंट नहीं है. सो दिस टाइप ऑफ द रेप्लिकेशन यू कैन कॉल इट एज ऑटोनॉमस रेप्लिकेशन. सो इट इज not dependent on main DNA for replication. It is not dependent. It is dependent नहीं है, खुद replication कर रहा है. That's why you can call this replication as autonomous replication. अब इस प्लाज्मिड के अंदर जो जीन्स प्रेजेंट है, they are not important for the growth and viability of bacteria. Very important point. प्लाज्मिड के अंदर जो जीन्स प्रेजेंट है, they are not important for the growth and viability of bacteria. मतलब कि अगर बैक्टीरिया के अंदर प्लाज्मिड एब्सेंट है तब भी बैक्टीरिया अपनी नॉर्मल एक्टिविटीज शो कर सकता है. बैक्टीरिया कैन सर्वाइव. सो इट मीन्स व्हाट यू आर गोइंग टू कॉल देम एज एसेंशियल और नॉन एसेंशियल एलिमेंट्स. बैक्टीरिया इसके बगैर भी अपनी नॉर्मल एक्टिविटीज कर सकता है, तो क्या कहेंगे आप इनको? नॉन एसेंशियल एलिमेंट्स, और यू कैन ऑल्सो कॉल देम एज dispensable elements. They are non-essential elements, or you can also call them as dispensable elements. Now, the plasmid in the bacterial cell, it can occur in two states. बैक्टीरिया के अंदर ये दो स्टेट्स में सरवाइव कर सकता है. वन इज फ्री स्टेट, द फ्री स्टेट which I have shown here. Yes, this free state of the plasmid, and the second state is integrated state. Let's see what is the integrated one. ये main DNA है, and in the main DNA now you can see there is integration of plasmid. This one is main DNA. This one is plasmid. Now the plasmid is not free. Plasmid is present in integrated condition. When the plasmid is present in integrated condition, this type of the plasmid, now it is known as episome. Integrated plasmid. What it is going to be known as? Episome. So this is all about the different characteristics. So this is all about the features of plasmid. Now I am going to deal with the types of plasmid in detail. So now I will be dealing with the types of the plasmid. First category which I will be dealing here, that is sex plasmid. Sex plasmid, also known as F plasmid. F is fertility plasmid. Let's say this is our bacterial cell. In a bacterial cell, this is main DNA. Along with the main DNA, there is presence of F plasmid. Yes, the plasmid which I am dealing here, that is F plasmid. जिस बैक्टीरिया के अंदर F plasmid होगा, उस बैक्टीरिया के अंदर बनेगा पिल्लाई. उस बैक्टीरिया के अंदर बनेगा पिल्लाई, and this pili which is formed here, this is known as sex pili. This formation of sex pili. The bacteria which contains F plasmid, this bacteria is known as F plus bacteria. The another name, it is known as male bacteria, or it is also known as donor bacteria, right? तो जिस बैक्टीरिया के अंदर F plasmid होगा, that is known as F plus male or donor bacteria. Now in this bacteria, this is main DNA. Yes, this is main DNA. But now the F plasmid is absent. F plasmid is absent, so the structure, that is the sex pili, is also not formed here. जिस बैक्टीरिया के अंदर F plasmid absent है, this is F minus bacteria. This is female bacteria or recipient bacteria. Got it? Let's see what is the function of the sex pili. Sex pili can join male and female bacteria by forming a tube-like structure. Yes, It can join an male and female cell by forming tube-like structure. This tube-like structure which is formed, this is conjugation tube. Now what is the function of the conjugation tube? Through the conjugation tube, genetic material is transferred from male cell to the female cell. Genetic material का ट्रांसफर होगा from male to the female cell. तो जेनेटिक मटेरियल का ट्रांसफर कौन से सेल से हो रहा है? मेल सेल से. दैट्स व्हाई द मेल सेल इज नोन एज डोनर सेल. और इस जेनेटिक मटेरियल को रिसीव कौन कर रहा है? फीमेल सेल. दैट्स व्हाई इट इज नोन एज रिसिपिएंट सेल, रिसीविंग पार्टनर. राइट? थर्ड कंडीशन. This is main DNA, and now F plasmid is present in integrated form. F plasmid is present, so surely there is formation of sex pili. Now this type of the cell where the F plasmid is present in integrated condition, it is known as HFR cell. HFR, please write the full form. High frequency recombinant cell. Yes, high frequency recombinant cell, also known as metamine or supermale. Now how this genetic material is transferred with the help of F plus and with the help of HFR cell, this we will deal in detail while studying reproduction. So this sex plasmid is clear. Now why it is known as the sex plasmid? Because it will determine the sex of organism. Plasmid hoga, so which sex? Male. Plasmid absent hai, so it is female. Now after the sex plasmid, next category I will be dealing here, that is degradative plasmid. To explain this, I will be citing one example here. Now the scientist who has worked with the degradative plasmid, that is Anand Mohan Chakravarti. Anand Mohan Chakravarti has formed a superbug, a bacteria that is Pseudomonas putida. Yes, he has formed a superbug that is Pseudomonas putida. इस Pseudomonas putida के अंदर आनंद मोहन चक्रवर्ती ने 4 डिफरेंट प्लाज्मिड इंटीग्रेट किए हैं. इस Pseudomonas putida के अंदर आनंद मोहन चक्रवर्ती ने 4 डिफरेंट प्लाज्मिड्स इंटीग्रेट करे हैं. ये जो 4 डिफरेंट प्लाज्मिड्स इंटीग्रेट करे हैं, they result in the formation of certain products. And what is the function of these products? These products, they can degrade. Yes, they can digest hydrocarbons present in petroleum. Yes. So these plasmids, they form certain products, and these products, they have the ability to digest hydrocarbons present in the petroleum. So it means Pseudomonas putida, it is a petroleum eater. Pseudomonas putida is a petroleum eater bacteria. It is a oil eating bacteria. Now, these 4 different plasmids, they have the ability to degrade 4 different types of hydrocarbons present in petroleum. 4 तरह के हाइड्रोकार्बन्स को डिग्रेड कर सकता है. दैट इज कैम्फर, xylene, octane and naphthalene. Right? तो अगर कहीं पर भी ऑयल पोल्यूशन होगा, पेट्रोलियम पोल्यूशन होगा, दैट पोल्यूशन कैन इजिली बी कंट्रोल्ड विद द हेल्प ऑफ सुडोमोनास ब्यूटिडा. यस, it can be easily utilized for the control of pollution, oil pollution. जब भी हम किसी माइक्रो ऑर्गेनिजम्स को, लिविंग ऑर्गेनिज्म को यूटिलाइज करते हैं for the control of pollution, this process, it is known as bioremediation. Yes, please write it down. Pseudomonas butida is involved in bioremediation. That is, utilization of living organism, utilization of microbes for controlling the pollution. And the pollution which we are dealing here, that is petroleum pollution, oil pollution. Now after degradative plasmid, now next category, that is called plasmid. जिस बैक्टीरिया के अंदर कॉल प्लाज्मिड होगा, दैट बैक्टीरिया विल फॉर्म टॉक्सिन्स. Name of the toxin is colicins or bacteriocins. Now these toxins which are formed by the bacteria, they will be released from the bacterial cell and they have the ability to kill sensitive bacteria. Right? So these toxins, they are secreted from the cell and they have the ability to kill those bacteria in which the called plasmid is absent. Right? Now the next one is R plasmid. इस R plasmid के अंदर present है RTF. RTF, that is resistance transfer factor. जिस बैक्टीरिया के अंदर ये प्लाज्मिड होगा, this bacteria will be resistant to antibiotics, UV rays and heavy metals. This plasmid will create resistance in the bacteria for the antibiotics, UV rays, harmful radiation as well as heavy metals. Now the last category of plasmid, that is TI plasmid. TI. T is tumor, I is inducing, that is tumor inducing plasmid. Right? This tumor inducing plasmid is present in bacteria, that is Agrobacterium tumefaciens. Present in Agrobacterium tumefaciens. Yes. Now this bacterium, Agrobacterium, is very common in soil. Yes. It is very common in the soil ecosystem. Let's say this is soil. In the soil, this plant is growing. And Agrobacterium tumefaciens, it is very well common in the soil ecosystem. Now this bacteria, it has natural capacity to attack the plant. इसकी natural ability है plant को attack करने की. ये plant को जब attack करेगा, इसके अंदर जो प्लाज्मिड प्रेजेंट है, टीआई प्लाज्मिड, वो प्लांट के अंदर ट्रांसफर हो जाएगा. प्लांट के अंदर ये टीआई प्लाज्मिड रिजल्ट्स इन ट्यूमरस ग्रोथ. यस, दिस रिजल्ट्स इन ट्यूमरस ग्रोथ इन द प्लांट लाइक दिस. यस, tumorous growth is resulted due to the TI plasmid, and this disease which is caused, it is known as crown gall disease. Are you getting this? Soil से बैक्टीरिया नेचुरली अटैक कर सकता है प्लांट को. प्लांट के अंदर टीआई प्लाज्मिड ट्रांसफर होगा, जिसकी वजह से प्लांट के अंदर होगी ट्यूमरस ग्रोथ, एंड द डिजीज विच इज कॉज दैट इज नोन एज क्राउन गॉल डिजीज. नाउ दिस बैक्टीरिया इज वेरी इंपॉर्टेंट बैक्टीरिया. You will study in the 12th standard. This bacteria is utilized for transfer of genes through the genetic engineering technique. So this bacteria is widely utilized in the genetic engineering for the transfer of genes, right? So this is all about the different types of the plasmids. Now after the plasmid, one important component of the cell which we will study here, that is the cell wall.